I got a point. I know.
Oh, 42.
I know. I know. Listen, I know.
That wasn't exact. You were off by 0.5.
0.5. Okay, final exam review. Very cool. So for chapter 2, mitosis, 3 is Mendelian inheritance and probability, because it has to do with like a hybrid cross. Or no? Yeah, a hybrid cross. 4 is sex and sex linkage. Qualifications to Mendelian inheritance, including epistasis, penetrance, and maternal effects. 6 is pedigree. 7 is linkage, including recombination, having to utilize match to do so. 8 is mutation and chromosomal variation, which occurs via non-disjunction. 10 is the DNA molecule. What is it?
11 is replication.
Is 11 lysosomal or is that a different chapter?
Yes, it is.
And then what, transcription? I think so.
I believe.
I think like 12, 13, 14 or something is it's at some of them it's
We have
like
it right there,
the replication, transcription, translation. I know which one is replication. I know replication was like the first one we
12
did,
is
but I don't
replication.
remember.
11 is chromosome structure, organelle DNA. Whatever that means.
Organelle DNA.
Yeah.
That is what makes the energy in your cells.
ATP.
The cellular respiration. What undergoes cellular respiration? What organ? Which organ?
Mitochondria.
Does mitochondria share our DNA?
It has
No.
maternal
As
DNA.
it's
Exactly. So, yeah, mitochondria have their own bacterial DNA that we inherit from our mothers. So I forgot what you said. Chromosome.
chromosome structure and organelle DNA.
Huh?
I only remember we talked about like the replication theories. Oh wait, I think we did that one online.
Transcription.
What are we transcribing? The DNA.
Sure.
DNA to RNA.
So we're transcribing DNA. There you go.
Yeah.
What's next?
Translation?
Should be translation.
It's like RNA molecules and RNA processing and then translation, but they kind of all just blend together
Yeah,
in my head.
with the codons and stuff.
Yeah.
14 is just RNA processing.
Then translation is 1.
15.
15 is
What?
14
translation.
is just processing. What are we translating?
RNA strip or processed RNA.
Then 18 is mutations, I think. Yeah, gene mutations and DNA repair.
Yeah, DNA.
What repairs are DNA?
Many things. Exonuclease activity.
What has exonuclease activity?
The DNA does.
DNA
polymerase
polymerase 3 and 1.
DNA polymerase does.
How does DNA polymerase 1 go about fixing its nucleotides?
1. Isn't it in the 3' to 5' direction? And then
And
polymerase 3 is 5'.
it goes
to 3'.
over, it
Or they go-
rereads the molecule.
So there's
to
no
fix.
5' and 3'.
Could your final answer?
Final answer.
Final answer.
Proofreading.
Let's see. I'm gonna say they're both 5' and 3'.
They're not both.
Yeah,
I thought one of them. Yeah, one.
They're
is
One
opposite.
is one
Yeah,
is
they're
one.
off.
why would they be opposite? How could that? Because it's one's gonna go one way and then the other. So one does have the 3 one has the backspace ability to like undo mistakes it makes, but then the other one, the DNA
One.
polymerase 1 is what removes the primers.
It's like a rerun.
And then
Yeah,
so
yeah,
it has
yeah,
to go the other way.
removes
So
the
that's going to
primers.
be the proofreading. So if DNA polymerase 3 is going backwards, it's backspacing. Which direction is backward?
5 to 3.
Yes. Good job. Thank you.
Right.
Yeah.
What's that?
I was just asking for DNA polymerase 3, right?
Yes.
And then DNA polymerase 1 is 3 to 5. Good question. I'll tell you why, actually. It's because it's reading the molecule to move the RNA primers.
Yeah, it's reading DNA in the proper or
Direction.
forward.
Proper. The proper direction.
The proper direction.
It is 25.
I just didn't like the way it looked. I don't like what that was.
25.
Be careful what you say.
We just did it.
We just did it.
That's the population
Population
genetics, right?
Perfect.
Yeah.
What's included in that?
Weinberg.
Hardy Weinberg. Equilibrium.
Yes, P's and Q's. I was hoping that you would just say Hardy Weinberg, because that's not the focus. The focus is population. Okay, very good. So how many is that?
I did. I was the first person to look.
I have a list
So you
of
only have
everybody.
15 chapters.
Only 15.
You're ready to go nervous.
Did I or did I not say happy birthday when she walked?
You didn't.
Is it your birthday?
It is my birthday.
Happy
Thank
birthday.
you.
I think we even started at 12:06.
I wish. Yeah, I think I walked in at 12:06. 12:06 is my favorite time ever.
That's what you need to know.
Beautiful. How many
Take a
chapters is that?
picture.
2, 4, 6, 8, 10,
15.
12.
Let's see.
It's 15 chapters, by the way. I just counted.
Thank you for learning.
My phone kit was having issues. There we go.
Yes, this is probably backwards, so whatever. Okay. This is what I like to do for my exams, to assess what I need to know in the first place. What did you do?
I took a picture.
I'm sorry.
No, all good.
So what I would like to do now is challenge you with some questions that assess your ability to answer questions regarding those concepts. Start from the beginning.
Alex, would you call him my incapacitated? I was like, no, not incapacitated. He's
Incapacitated.
crippled, maybe.
Wheelchair.
Wheelchair.
You can go as slow or as fast with all of these questions as you want. We can skip questions if you want, but there might be some that I would like to focus on that I know that will be presented to you.
Do we do them together or do we write it down on our paper and then discuss? Are we discussing first or what's the vibe?
Together.
Together,
You
okay.
might have the answers, but one of your friends might not.
Sure.
Bring each other's thought process is what's important. So that we can let us know the answer.
Okay. For, um,
I'm thinking replicated DNA goes with synthesis. Because that's the goal of DNA
True.
synthesis is to replicate the DNA.
So replicated DNA synthesis, so same things, E goes with gamma.
C and D, so C would be.
I think it's the delta.
Yeah, delta. Does M mean
Mitosis.
Oh, never mind.
mitosis? And
so I
what does
think
I mean?
cytokinesis.
Meiosis 1, meiosis 2, mitosis, mitosis. Could you repeat that, please?
I think cytokinesis M from mitosis is 2 identical daughter cells.
Cytokinesis M.
I
for
agree.
mitosis
Yeah.
is 2 identical.
And the other one is the other one.
Yeah,
the
the other
daughter cells.
cytokinesis, yeah.
D goes with delta and C goes with what?
The last one. I don't
The
know what
E.
the one that looks like E.
Epsilon.
Epsilon.
So it is E.
And then CD.
Condensed chromosomes happen at prophase, I think.
Why?
Because that's what they have to do in order to prepare for separation, right?
Yes.
And then alpha.
Yes. And then for that. So B goes with alpha and A goes with beta. So just to pose an extra question on top of what Sid said, why do we condense our chromosomes for prophase? Or no, you answered that, sorry. But what kind of DNA do we have before we condense our chromosomes?
Oh, is it the not supercoiled? Do we supercoil it or is it start as supercoiled? I don't
When do you
remember.
find supercoiled DNA?
I don't remember.
Isn't it always?
In chromosomes.
So would it
Chromosomes.
become supercoiled to condense them?
Yes, so when I could pull that up, I've shown you before. You know, DNA chromosome, if you zoom in, is just a bunch of supercoiled DNA around histones in units that we call nucleosomes. And if you acetylate, is that right? Yeah. Then you unravel the DNA into a visible helix, double helix. So why do we have to? No, that's not the right question. What kind of DNA is it when it's supercoiled like that, when we condense it? It starts with a H.
Heterochromatin.
Heterochromatin, exactly. So we have heterochromatin, which is packed down really tightly so that it can move really easily through mitosis and meiosis and not get lost. But before that, before we condense it, before prophase, what kind of chromatin is that?
Euchromatin.
Yeah. What does U mean? It's a Greek prefix.
No clue.
What are other changes over here?
U means true. So this is the true chromatin that we can actually like get at. Our replication can happen there. Translation, transcription, and all that stuff. So yeah. I mean, why did you say that B goes with alpha?
Because in anaphase 1, homologous chromosomes separate, and then in anaphase 2, sister chromatids separate.
Exactly.
Right? Okay, good.
Perfect.
You're actually not allowed to say that I'm wrong.
It's your birthday.
It is my
Yeah.
birthday.
Actually, do you want to?
Oh, no, I'm not that confident.
Yes, so that's a very good point that we're going to end up doing it in a later question, but the purpose of meiosis 1 is to separate our sister chromatids from each other, or homologous pairs from each other, and then meiosis 2 is about separating those sisters from each other, because we're just trying to break it down into more and more simple parts, but in meiosis 1, we're still at the point where we have 2 chromosomes that represent all of our genetic information.
And then anaphase 2 of meiosis is the same as anaphase in mitosis.
Which meiosis?
No, anaphase. Anaphase 2
Oh,
of
yeah,
meiosis.
sorry. I think of it as anaphase of meiosis 2, so I didn't understand.
Oh,
Yes,
anaphase
you're right.
Yeah, and
Because
mitosis.
by the point of meiosis 2, our genes, our chromosomes are simple enough to, or such that like the only way we can further break them down is by splitting the sisters apart. And that's what we're doing in mitosis.
Splitting the sisters apart.
Any questions? I'm a zygote that will grow into an owl. How was I made? How much genetic material am I made of?
What are the parameters of
Okay,
this
so
question?
it's a zygote, so it has mom and dad's stuff, because it's a zygote, right?
It's fertilized.
Yeah, because I think a gamete is just one.
How was I made? Yes, we have 2 parents, female, male parent, mother and father. They give us something.
Gametes?
Yeah, they give us our gametes. How much genetic material is in gametes?
Half.
And
Or n, yeah.
You are both correct, half or n, because we expect an owl. Because we expect an owl, who is an animal, they're a vertebrate, to have a normal karyotype of 2n, or
Karyotype.
no, the karyotype one. Actually, I think owl's karyotype is like 82. Maybe it's 86.
I'll look it up.
I think it's 82.
Final answer.
82. Yeah, but their ploidy is 2n because they have. What does it mean to be 2n?
Diploid.
Yes. And what does diploid mean?
You have 2 copies for
So yeah, we know di is the Greek prefix meaning 2. What does ploid mean?
Or no.
What did you say your answer was?
82.
It is 82. Yes.
What is a ploid? It's Greek. Where does Greek people think? What's it? That's a great guess. That's much closer than you think. Yes, it is referring to chromosomes because ploid means fold. And chromosome, when we think of it, it's this guy, it's kind of folded up, wonky. So diploid, 2 folds, they have 2 chromosomes that represent their genetic information for a particular chromosome. Owls have 41 of those. So many.
No. It's okay. I got a question.
Okay. So that's how much genetic material. Oh, firstly, so in owl, we expect them to be 2n. So by this point, the genetic information that their parents pass on is haploid, as Sid said. So this n is haploid. When these two combine, what is n plus n?
2n.
Hooray. And this is diploid. So that's how they were made, and that's how much genetic material. Sick. We went through all the chapters and
I took a picture.
decided what we haven't gone through all the chapters. We just,
Okay.
said what the concepts are in the chapter. Okay, perfect. And now we're going through some questions, and we can go through them as fast or slow as you want. We can skip any that you don't want to do because there are like the semester's worth of questions there.
Okay.
Well, this is a good one.
Is it online? Or is this just?
This is in my Google Drive. I think I may have given access.
Yeah.
I can make this a podcast.
Yeah, no worries. It is podcast assignment is due today for extra credit.
Mine. I like it.
It doesn't take that long.
Yeah.
They're so interesting. Like, there's pages and pages. I started with one and attempted to do so many more. I started with the runners one because I'm a runner. So it's something that I found was really like the sport gene series. There's 2 and I was not athletic until much later in my life. And play sports. So it's just interesting.
You'll have to tell me about that. I've never heard of that.
Okay. Skin cell. For sure.
Which of the following cells would you expect? Okay, my test is. Oh, I gave you. Sorry. So we think the skin cell. What?
Somatic.
2n.
Yeah.
Not the germ cell.
Why not? What's a germ cell?
1n.
It's sex cell.
It's a game.
You all gave different answers.
They're all the
They
same.
all mean
No,
the same
yeah, they're all
thing.
contributing the same answer.
Synonyms.
Yeah.
They do not all mean the same thing.
Oh.
You said gamete. You said.
Game.
Oh,
I
thought when you
said sex cell.
But
I
you
said
said sex cell,
She said
haploid,
haploid.
and gamete.
All kinds
So
of
3 of those are the same thing.
Who's wrong? It's me?
Oh, it's not a gamete.
No, gamete and sex cell, definitely the same thing. Haploid, definitely.
I said n is
yeah.
haploid, yeah. So is it not n?
Yeah, it definitely is N. You're definitely
If
right.
it's haploid?
It's definitely a gamete.
So we make gametes. What process?
Meiosis.
Meiosis.
So the end result of meiosis,
Or
Please
unique
spell
daughter cells.
meiosis, the result is 4 times n, or haploid gametes. Where do they come from?
Where do gametes come from?
Oh,
This process,
Yeah, they come from meiosis.
meiosis happens, but where do they come from?
The existing cells from the parents.
And what are those existing cells called?
Germ
Germ
cells.
What's another name for germ cell?
Sexa?
Be careful with your gametes, your germ cells, and your sex cells, because we start off with germ cells that are diploid,
Oh,
and we then reduce them via meiosis. Meiosis 1, we break apart the homologous chromosomes so they are now N, and then in meiosis 2, we break the chromosomes apart into their constituent sister chromatids to make them able to combine with another organism's single sister chromatid. That's how we have it before.
To reiterate, a sex cell or slash germ cell is 2N? Okay, so we would say check for this question, like it's part of the.
but it still doesn't go through mitosis. It goes through meiosis.
Okay, gotcha.
You're born with all the egg cells you'll ever have in your life. And born with all of the sperm cells I'll ever, no, the sperm progenitor cells that I'll have in my life. They undergo meiosis constantly to produce new gametes. Except you. Never mind, that's a can of worms. So germ cells do this, which is not this. Long story short. Sorry, that was a good idea.
So it does not go mitosis, and the reasoning behind it would be because it's 2N, but it has to undergo meiosis instead
To make it.
to make
Yeah, to make
gametes.
That's its whole purpose.
Because you have
Okay,
to,
so it's still, it would be X.
you have to start with 2N to make your N.
Yeah.
Oh, you don't have to take yourself away from the kidney. So not this one.
Yeah.
No.
What about this one?
I don't know what
Is that a
mycelium
yes?
is. I think it's a yes.
Mycelium.
Like if this was the exam, I'd check it.
Because I'm
Yeah.
thinking about, 'cause we did something with some kind of thing like that, what we did in lab with the, what you call it, I don't remember what they're called, the
Sordaria?
Sardaria, yeah,
Fungi,
those guys.
that's a fungi.
The fungi, yeah. So I think,
They're really
but
pretty.
they're fun guys.
This was a trick.
Ah,
It's not a check. Oh my god. So many folders.
biodiversity. Learned this at some point in my life. I don't remember anything, but
Feels like ages ago. COVID, learning and COVID.
it didn't feel real.
So
Oh.
this was a trick question because the city of my seats at the least, or no, these are Oscar my seats, at the very least, had this what's called an extended dikaryotic phase, where they have two ends, two sister chromatids that don't join. They remain in the same cell, but they don't combine. So they're N plus N rather than 2N, as trying to trick you.
Mm.
So the answer is
Hey, so if you go back to the to review. Thank you. Thank you. So this was a trick question. You will never be asked about that.
Oh.
But the answer is no, because it has this extended dikaryotic phase. Dikaryote, like prokaryote or eukaryote, die because it has two.
Hold on, wait for it, wait for it.
Two individual chromosomes from a parent within the same cell.
Was that how it was with the Sargaria also? Because I know we had to, when we were doing some kind of calculation for it, we had to also, I think it was genetic mapping recombination, we also did have to divide by 2. Because there was something like that.
I do not remember.
That's how it is.
So yeah, that's a trick. Don't worry about it. What about a human's neuron?
Don't
Yeah.
think so. Is it a cell? Is it a
Cell.
neuron?
It is a cell,
Neuron?
it is somatic, but I don't think they replicate.
Why?
I think it's.
I'm a neuroscience major. I feel like my words. should just
I
be
know.
okay. Oh,
Yeah.
yes.
You are correct. But concerning the cell cycle, why?
Concerning the cell cycle,
Because we know that we have our cell cycle. Any particular cell will go about it or stay in different stages for different periods of time. We usually have our G1, our synthesis phase, our G2 phase. We also have a G0 before we've started G1. And then we have a short window for mitosis. Some cells will undergo meiosis. And then the cycle will reiterate. Things like liver cells or your skin cells will do a lot of this because they have to constantly divide. So they will do a lot of this and a lot of this. But things like our neurons, where do they fit into this?
they're in the genome. Really? Forever?
Are we born with all of our?
So there's a trick to that. In your nose, so most of our neurons cannot replicate.
Mhm.
That's why neurological damage is so harmful to organisms because it can't be repaired. But in our nose, at our olfactory bulb, which is our first cranial nerve, that there's an upregulation of some chemical, I can't remember the name of that, like allows your nasal or your olfactory nerves to replicate themselves. So that's actually like a therapeutic technique that's been innovated in the last few years, that people will have transplants of their nose nerves. It'll get put into their bone marrow. And those will be new stem cells so they can grow new
Really
tissues.
cool.
There used to be a like a neuron simulating like a museum exhibition, I think, in DC that I was just listening to the NPR, and like it walks you through the colorful life of a neuron all the way from like the beginning to.
We should go there. Field trip.
That's
Yeah.
that'd be awesome. Yeah, it's really cool.
What's that? Is it still there?
I think it might have
Name?
been a pop-up.
I don't want to know.
What's the name?
It's just at the Natural History Museum history.
Where you are?
This girl.
Field trip.
Field trip.
Except it costs like $10 to ride the metro.
Okay,
If you go on the
okay,
weekend,
not
it's
that
not.
much.
On Sunday they had the
Weekends, it's
the,
$2.
thing that they had.
Oh, on the weekends.
Weekends, it's always $2. And then the buses are sometimes free on the
Isn't
weekends
there like
also.
I feel like don't we have a discount for students or something? Like locally, I think I know we do, but it's hard out there, man.
It's hard out here. Okay, so neuron, like Jazz said, is usually stuck in the genome phase. There are examples, but if we're stuck here, then we're not gonna. It's a good thing.
True. Okay, a bee's exoskeleton. Yeah, it does.
Why?
Don't know, but
Those
They're
are also,
always
those are
like-
cells.
exoskeletons.
Yeah, they
They repair,
sometimes,
they repair
sometimes.
this
They-
stuff,
yeah,
right?
they
I'm
do repair
thinking,
themselves,
I'm thinking
yeah.
of like how lobsters work, which is not like bees
How lo-
exoskeleton.
That would be a good band.
But it's like because lobsters they grow their second shell underneath the first one and then just break open the other one. And it takes a while to harden, but like that has to be like they have to be producing and replicating those cells to make it.
process.
That's my process of thought.
We're talking about the process of ecdysis because all arthropods are ecdysozoans.
Mhm.
But
I
this
don't know
is
about
another
bees,
trick
so.
question. I'm sorry. What actually happens is that not just arthropods, but many organisms have a basal membrane with columnar epithelial cells, and these epithelial cells secrete a waxy
Ah.
cuticle, which can then sclerotize, harden, and become a nice protective exoskeleton. So the exoskeleton itself is not even a cell. This was another trick question.
Oh, wow.
So there's only one answer to this.
So what if this was the exoskeleton and it was just some really tough cells.
Yeah.
Then yes, it would be capable.
But wait, why isn't the exoskeleton able to do mitosis?
It's
Not
not a cell.
cells.
Good job, team. We didn't do this in this class. This was in Dr. Kara's class. Could you scroll down, please? Okay, perfect. You're like,
Okay.
an organism has 2n equals 14 chromosomes. How many chromosomes and chromatids will be present during prophase of mitosis? And I am willing to draw out the cell cycle phases. This is reversed. I think it's probably reversed.
Yeah, I got this wrong on the first test.
It's gonna be the same
I'm
number,
tired.
well, right?
Seven is
They haven't
every
separated
single-
yet. It's gonna be the same number of chromosomes and chromatids because they haven't separated yet.
Huh?
I'm just throwing it out there.
Yeah,
I don't
yeah,
know
they
which
haven't-
number, but I
that's
think
right,
they
they
haven't separated.
they haven't
so
separated.
each count as
They have
one.
duplicated.
That's
Yeah.
That's a great point and an excellent way to approach any question like this. If we want to know what's going on during prophase of mitosis, then we should like, look at what's
I
happening
agree.
in prophase of mitosis by drawing.
The chromosomes will be the same number, but chromatids will not.
So what happens in prophase that we saw from that first question about matching?
Prophase,
They're all getting ready. They're
they're condensed.
condensing and they're there.
So prophase is where our nuclear envelope, you don't need to know that part, but it's where our chromosomes condense, or our DNA condenses into chromosomes. So all the genetic material that we have, this cell is typically a 2n equals 14 cell. I don't want to draw 14 in this. It will be present here. It's just condensed right now. That was my.
So it'd be A.
Yeah,
it's
I think
14
A.
chromosomes with
28 chromatids.
Why not B?
Why not B?
Oh yeah, 'cause the chromatids are,
Because we've duplicated
yeah.
When?
in synthesis.
Yes, perfect. We, before prophase, because prophase is simply a part of mitosis. Before that happened, we had our G0 and our G1 and our S phase. It's during this S phase that we have the contents of chapter 12, which is DNA replication, where a single sister chromatid will become duplicated. So what was previously a single DNA molecule or chromatid has now created a copy of itself. And now every chromosome is comprised of 2 sister chromatids or DNA molecules. I prefer to say sister chromatids because it makes me sound cool.
Huh? Maybe.
I don't know.
Okay.
So what is A? Yes.
I think so.
It's back from the other side.
Poetry yours.
It's a nice turn. It's raining outside and that makes me.
Oh yeah, it's drizzling out there.
Such a gloomy day.
Perfect for studying.
Absolutely.
Take a nap.
Yeah.
We can have nap time,
It was hard to wake up today.
but it's the best day of the year.
It is the best day of the year.
Okay, so could someone explain to me again why it's A very nicely and concisely that right?
Because we've already gone through S phase, our chromatids have made sex. We have 2 chromosomes, 2 sister chromatids. And because we're 2n, we have 14. Because they're now condensing, separating into chromosomes.
Yes. So why don't we just look at this as an equation and say, oh, n equals,
Like n equals 7.
or
That
even
would be meiosis. Kind of. Not 100%, but that's closer to meiosis than, because we end up with the N in meiosis. We don't do that in mitosis.
Okay, yeah, that's a good point. So what I was getting at is that this is not an equation. You must never try to solve this as if it were an equation. This is simply a sentence that says an organism has 2 chromosomes forever. 2 chromosomes represent the genetic information for every like, chromosome unit. You know, we have 23. We have 2 chromosomes that represent each of the 23 chromosome types. And there are 14 total. This is not an equation. This is simply saying that. So yeah, the process of what you said and what you all did, finding that it's 28 is good. Think of this cycle.
I'm sorry.
Thank you,
I
Jen.
get to scroll.
Yes.
The answer is A, right?
Yes.
Okay.
Would this one be D because we're ending with a haploid cell, we started with a diploid cell.
Explain, please.
If the daughter cell is at the end of mitosis too, so
Yeah.
that's haploid, that's n
Yes.
equals 56. So then if you're a diploid cell, it would be, I know we're not supposed to do equations, but like it essentially it's like you're
It's
having
double,
2
yeah.
of those,
Yeah.
you're doubled the amount, so it'd be the 2n equals 112.
I
You're not
agree.
wrong.
It's applying the concept.
Define the concept.
Because it's a haploid plus a haploid equals
Yeah.
a diploid.
Do we want to go through it? Do we want to try? Would that benefit anyone?
I think I'm good. Can we talk about it if it were meiosis 1?
Yes.
Like a different,
Oh,
Then it would still be 56, right? Or maybe not, I don't remember.
that's where,
Yeah, because I don't remember if at the end of meiosis 1, I think the end of meiosis 1, you were haploid, but then that's why, maybe, I don't remember.
no,
It's something
No,
that
you're
you
still
continue to
diploid.
be.
I think you're
After
still
meiosis.
diploid, yeah.
You're still diploid and then you become
Because
haploid.
your homologous chromosomes.
It's been a while since meiosis.
I
Don't
remember- I
giggle.
remember this conversation like 12 weeks ago.
12 weeks ago.
So it seems to me like we should try.
Yeah,
Yeah.
Can we
Can
make
we-
it a
can we-
smaller one,
yeah,
like not 112?
Yeah.
Absolutely, yeah. Let's just do 4. I don't want to draw it, actually.
let's do
So I'll
2.
just I'll use numbers to represent it, and it'll still be 56.
Oh, okay.
Is that okay?
yeah.
Jenna,
that's
bring it
cool.
to-
Use the
Yeah, we'll
numbers.
use numbers.
Okay, so if we have 56, oh, you have to work backward.
Yeah,
Yeah, in meiosis 1.
Okay, so meiosis 2. Let's just articulate how to get to meiosis 2. So let's say we have meiosis 1 and meiosis 2. It's the first stage of meiosis 1.
prophase.
Perfect.
What is the N and P net?
Metaphase.
What about the T?
Yeah.
Telophase.
Telophase.
What about the C?
Cytokinesis.
That's wrong.
Looks like a butt.
Yeah.
So 56 in each.
There are 56 of these.
Yeah.
That's something we should always take care of. And there are also 56 of these. What's going on in here?
That's when they're separated.
The
There's the
squigglies are
100.
each side.
What's on this side?
It's 56 on each side.
Okay,
56 of
this
what?
is good. This is the
So
squiggly.
it's squid-
Yeah.
How many squiggles?
it's one on each side.
Singular.
One on each side.
Yeah.
What about this?
Squiggly is good.
56.
They're being separated.
112. Yeah. No, not 112, sorry.
No,
Actually, no, you're-
56
Yeah, I think it
X
is.
down the middle.
That'd be metaphase.
Yeah.
Are we not doing
56.
it?
So they're
Oh, they're like midway.
They're being
They're already
pulled,
torn
so the-
apart.
they're
Yeah,
like-
they're
But
It's
like-
still
torn.
56 on each
but
side.
it is still 56, yeah.
Because they're being
Damn,
torn
I need to draw
apart.
this out at the beginning.
spindle fiber.
So it is 112?
I think so.
So
I think so.
No, we'll
About.
It's one-
we'll have 56
Yeah, it's
chro-
112.
we'll have
It's
56
112, it's
chromosomes
still
and we'll have
Metaphase is
112
one
DNA.
phase.
Yeah,
so
yeah.
metaphase would be
Or
56
56 of the
Of the-
chromosomes.
because they're not separated yet.
Before we talk about that, obviously there's confusion about what happens in each of the stages. So what does
You don't-
what does prophase even mean? What does pro mean?
Before.
before, so it's what happens before stuff. Yeah, it's preparation. It's what happens before anything splits,
Like a
which is the true
prologue.
goal of meiosis. So prophase is what happens before any splitting. What is metaphase? What does meta mean?
Middle.
Middle.
They're lining up in the middle.
Exactly, chromosomes align in the middle. What does anaphase mean?
Separate.
Separate. Where else have you seen that word in your, or this prefix in your biology career? Yes, actually,
Yeah,
yes.
that's really good, yeah.
And I think Toby is, I actually don't know. So, but Anna, if you like break down the parts of your body, that's anatomy. You study that.
What was the word that you were
Oh.
thinking of?
I was thinking of anabolic reactions.
Oh. Okay,
What
that
is an
makes sense.
anabolic
Yeah.
reaction?
We learned that.
Actually,
we
that's
learned that.
backward.
It is because catabolic is the
Yeah.
destroy one.
Because a cat knocks the
Yeah.
face off of the counter and it breaks.
Yes.
So never mind.
Yeah,
But Anna, for this purpose, means to break apart.
I know for hair growth phases, anagen is the
I like
growth
that.
phase also. So I don't, for hair growth phases, anagen is the growth phase. So I keep writing anaphase on my lab reports.
Do you need to know that for forensics?
Yeah.
Ooh, forensics.
What is telophase? What does telo mean?
The
Telephone.
end.
I could say, yeah.
Because that's with the telomeres.
Oh, yeah.
That's how I remember it, because we talked about it in the last review session. Because telomeres are the ends.
What does cytokinesis mean?
Cyto is cytoplasm, and then kinesis, I guess, is to split. Those are
so
cytoplasm.
Cyto is the cytoplasm or the cell that contains it.
Yeah.
Kinesis, like kinetic, it's movement.
Mm.
So it's when two cells
Move
move
to
apart. So knowing this and always keeping it in mind, this should be much easier. Metaphase, they align in the center.
Yes.
Yes.
How many?
As
56
chromosomes.
56 X's,
Because they haven't been
56
ripped
100.
apart yet.
112
It'd still be
chromatids.
112.
Yes.
Okay. I'm really terrible at it.
We aren't squigglies, we're still together.
Yes.
Two squigglies.
We're too squiggly.
Two squiggly.
So I mess up with I'm sorry, this is a separate question, but where it's like, where did the non-disjunction occur?
Mm,
Yeah.
Yeah.
that
We
one
will
was
get
not
there.
Okay.
Is that?
Where they stay
It's a
together,
big-
they don't
or they-
separate.
Yeah, it's like the which one? Which one did they do that? with?
Yeah, I'm like, I'm sorry, I already forgot PMAT.
Yeah,
What is pro?
before getting ready.
Well, it's all over the place.
So what is this?
56
X's.
squigglies.
X.
X's.
Oh, I thought that was the
Yeah,
X
'cause
was
we've
already
already
written
got-
there.
we've already got-
This is another thing that Sid said, but I want to make sure it's iterated or reiterated. Where are these?
Center
In the
How do they
nucleus.
align?
in just one line.
Why?
Because the homologous pairs have already separated. Now we're separating the sister chromatids. So there's only
Yes.
can only be one on each
Oh.
side.
Our cells know to place chromosome 3 in a certain spot. They know to place chromosome 24 in a certain spot. Well, we don't. So they will always align where they're meant to, but they don't have a partner to pair up with because in meiosis 1, partner was taken away.
They're lonely,
They know.
and they're about to be even more lonely. They get ripped apart.
What was that?
They got divorced. It was very
They
rapid
get
divorce.
divorced.
So these are in the center, single file specifically. Like Sid said. Where are we here?
6 X's.
Moving back into meiosis 1, we have 56.
And both
Chromosome.
of them.
How would you describe this chromosome?
What do you mean?
Well, the way I describe it is like if you had to maybe write a short answer about this,
Okay.
I would write it as there are 56 chromosomes where each chromosome is made up of 2 sister chromatids. So that's how I describe it.
Oh,
Or 2 DNA molecules. you could say.
and each one has a year.
Those wouldn't be considered daughter. Would they be daughter cell? I guess technically yes, but yes. But you just have to be careful with how many sister chromatids. So in meiosis 1, there are two pairs of 2 sisters, whereas in the last one, in meiosis 2, there's only one.
So
One chromatid.
let's say you were given a question.
I'm so sorry. I know this is like simple, but I will freak, like,
You're asking.
during a
perfect
test, I'll
questions.
freak out and
I'm very
forget.
glad you're asking these questions. So let's say you were told that you're given a question. You have two daughter cells from two different processes. So you have one process where you end up with two daughter cells and another process where you also end up with two daughter cells. You're told that one of them gives unique daughter cells and one gives identical daughter cells, but they still both give two. Which process belongs to which?
Well, the unique goes to meiosis and
Meiosis what?
Two.
One.
One.
Because
Yeah,
you
mine
only
also-
have two daughter cells.
Yeah,
Oh.
meiosis two is gonna be identical because they're the connected ones.
Big brain.
Yeah.
Wait, the question was, which one has the unique, which one has the identical?
identical.
So
is like
there is, let's say the question is asking, you have a genetic process or a cellular process that results in two daughter cells.
Yeah.
If one process created unique daughter cells, two unique daughter cells, and another created two identical daughter cells, which daughter cells came from which process?
So after meiosis one, they're still unique.
What happens in meiosis?
Random assortment.
Perfect.
Yeah,
that's true. Absolutely, independent assortment. Perfect. And that looks like, I want to draw it out. Let's say that you had a red chromosome here and a blue chromosome here. They both are homologous to each other. Red is homologous to blue, blue to red. And they both represent chromosome 14, let's say. If blue was here and red was here, blue was pulled that way, red was pulled that way. Blue would end up here and red would end up here.
Yeah.
But what if red and blue are switched, but none of the other 23 chromosomes were? Then red would end up here, blue here. That is variation in itself. Because we have to consider this, these will end up being split up so that this blue goes to combine with some other organism's chromosome or giving this red one.
Yeah.
So these, having a red versus blue, that's genetic variation inherent to metaphase. Yeah, no, not metaphase, meiosis. There's another thing that happens in metaphase. So yes, independent assortment happens in metaphase.
Does crossing over also happen in metaphase? I always mix that up. I always keep thinking like it happens in prophase,
I do
but
too.
it doesn't happen in prophase.
No, just for today. I'm usually not this nice to him.
Yes, crossing over in metaphase. Why in metaphase?
Because I'm assuming it's because they're all lined up next to each other, so there's opportunity for them to overlap.
It's easy to remember.
Yeah.
In prophase, they're like jumbled up.
They're
so
everywhere.
Only in metaphase when they have the opportunity to cross over. with their friends, what do they do?
Because they're already separating, so it's like, perfect timing.
Exactly, might as well just
Might as
swapping
well.
gifts as we go, yeah.
Bye, guys.
Happy birthday.
That's
Okay.
um,
There's gonna be
Sorry,
Six
six X's on each.
she didn't know. 56 X's. How about this?
still that, but they're just moving to be separated.
How about this?
56 homologous pairs.
112 X's
Yeah.
right down the middle. There's two.
Because the 56 and 56
Yeah.
are separated, then you move them back.
But if they're, but homologous pairs are, would be 2 chromosomes of each. would be-
I think that I remember like, cause like, if they're the X's, they still count as one. So like in the second one, when they're separated into just the two squiggles, that's when they start counting as separate ones. But since we have like 2 X's, they're still each one instead of, so when you separate them, they still are 2.
Okay.
And you put them together, they're still 2.
Why isn't this 2 chromosomes?
there's
That's
potentially
just-
there.
Because they have the same
They sure
genetic
have.
information.
There we
I
go.
remember learning.
That's true, like when
Okay.
you're looking at it, but the reason that we don't, like the conceptual reason that we don't consider this two chromosomes, even though each one individually will eventually be a chromosome in its own right, is because these are identical to each other. They're sister chromatids. This one was replicated during S phase DNA replication to be identical to this one. So they don't give it, this one doesn't give any new information that this one doesn't have. So there's no new instructions for our cells to use, so we consider it the same. It's just a single chromosome because it only gives one unique set of instructions.
That's the coffee.
I remember you saying, ask me that question at like 10 p.m. in that one review session, and it just never left.
It's stuck forever.
Okay, so how many chromosomes are in this cell? Perfect. I'm so glad you said that because these cells haven't separated yet. There's still 112. It's at this point, cytokinesis, that each cell only contains 56. Perfect. I think I drew that out a bit longer than I should have. I'm sorry. Perfect. I will stay as long as you guys. I don't have anything. Well, I do. But so that's it for this document.
Okay.
Not the big red. If you could click out of that. Go to 300.
Are you gonna share this stuff with us?
Yes.
Okay.
And then
I do have to do it.
spring 22,
So is Menard doing something today or tomorrow?
4 to 5.
For-
It'll be online and recorded.
Okay.
This is also being recorded. I don't know how valuable it is. Okay, so I have literally 15 review documents to go through. You guys choose which week you want to talk about. Which topic, and I think I remember which week that corresponds to.
I'm trying to remember the first few, like on our first exam. Oh, I think I took a picture.
Okay.
Like some of the bigger questions.
It's been a while. Hold on, sorry.
Questions about mitosis and meiosis?
No.
So, okay.
Non-disjunction is my metaphase? Or,
It can be any time.
oh, really? Okay.
Well, it happens as a, so glad you did the effort to be non-disjunction.
Oh, yeah. Camera can see the way I look.
Oh, that's non-disjunction. What does it mean?
Not. Junction is a connection.
And this is like none or like the opposite.
Separate?
Separate. It's something.
Yes.
Disagree is just like no. So you don't agree. So it's like something like that.
So in a junction is two things together. Disjunction is a separation of the two things from each other. In our cells, we want this to happen in anaphase. We want our homologous chromosomes to separate from each other. We want to undo their junction. And in anaphase of mitosis and meiosis 2, we want the sister chromatids to separate from each other. But when that doesn't happen, when we don't separate our chromosomal units like we want, that's non-disjunction, and that causes some funky business.
Okay, question. Non-disjunction can occur in mitosis, but it's more bad, so those cells die. So the ones that live is meiosis because we can counteract that. Or you can live with those changes, correct?
Yeah, our
Okay.
somatic cells, like we have so many billions of somatic cells, and they're going to be replaced, hopefully. So if one of them, the ones that undergo a lot of mitosis, they'll get, if they have something wrong with them, they'll die and easily be replaced. But our germ cells often were born with the only ones that will ever have. So,
If they,
wait, that's not a good connection. The connection is that one of the non-disjunction event in your your germ cells, like through meiosis, that could present or provide a gamete with problems. And if that one gamete happens to be the one that fertilizes or is fertilized, then that's an entire organism who only has that non-disjunct DNA to work with. So it could be really, really bad in meiosis for the offspring, but it's not as important in mitosis for the parental organism. I had a question. Yeah, you're welcome. Thank you for coming. What do you guys want to talk about? I have questions for days.
Do you mind if we go over, and I know we literally just went over this,
Do you wanna
but,
go over an example?
oh yeah, I guess I could pull it up. There are a couple questions I got wrong on the first and second.
Are you okay?
I'm okay.
I'm sorry.
It jumped out.
See, or if we can also go over, I know we just learned this, but I mix up, yeah, 25.
Perfect, no problem.
Sometimes I mix up like I know when I'm like how to do it. It's just, I mix it up occasionally.
Sid wasn't there, Bernard wasn't there.
But weren't we there?
The first day for chapter 25.
Oh, yeah.
And that was a really not good day to us. Because like she talked, like she made Hardy Weinberg
Yes.
really comprehensible.
Mhm.
I think maybe I think I was there. I remember learning. I know I was outside. I remember learning about
You were there
Pythagoras.
in the second.
Was that
But
less
the-
terrible?
Well, the first day like laid the foundation so that
Yeah.
you could really easily understand it. And so
Okay.
that you can not just like learn or know this is this because it is, but
Yeah.
know why.
Okay.
Okay, so I know we didn't talk too much about the, what is it called? The Harvey what equilibrium?
Hardy Weinberg.
Hardy Weinberg equilibrium. It's p squared plus 2pq plus q squared equals 1. Okay. I guess
That's
what
not
I'm,
the most important part. Sorry.
it's not, but sometimes I will mix up when I'm reading it. It's like I don't understand. It'll say this is the frequency in which this occurs, and I will forget to take the square root to get p and q. I guess like I understand what the allelic frequency is and what the genotypic frequency is. I just, when I start to do these problems, I get nervous and I can like
That is the trick to Hardy Weinberg. It's the only trick.
I'm gonna open lockdown while we're talking about this one.
Yeah, that's the trick. And I definitely, it's easy to mix them up.
And I'm sorry if you guys already went over this. I just,
It's always good practice regardless.
We're not
Yeah.
the only person who has problems with chapter.
like I understand it, and for the most part, when I'm like working through it in class, I'm like, oh, I got this. But I know test day, I will look at it and think to myself, is this right? I need to take the square root.
Just to start out. Now we can talk about that's fine. Soon humans can have compressed noses. My partner from last year, Sam, wrote this question. So, assume that humans can have compressed faces, and it's expressed as an autosomal recessive trait. So, actually, I'll read it first. A sample was taken and found that in a population of 300 people, 81 had this condition. Find the allele and the genotype frequencies of the individuals in this population. So this is already a hint. When it says autosomal recessive trait, what is that?
It's autosomal recessive.
You're not wrong.
It needs two of the same allele to express the trait.
Yes. You need to be what kind of genotype?
Homozygous recessive.
There we go. So this condition only is expressed in individuals with a homozygous recessive genotype at this locus. It's the P locus. And therefore, people who are either homozygous dominant or heterozygous. There's no distinction between parents and those letters. I'll do N for nose.
Okay.
And then heterozygous. These people have it, these people don't show it. These people are carriers, these people are not. So we found the genotypes. The thing about Hardy-Weinberg is what we're doing with Hardy-Weinberg is assuming that there's a dichotomy of what we're looking at. We're first looking at what we see and then trying to compare it to like an ideal situation where the allele frequencies in a population are static. But you know, evolution, nothing's ever static. So what we do is compare what we see to what is an ideal situation. And if they're the same, then we can say they're in Hardy-Weinberg, but if they're not, then they're not. So the first thing I like to do when it comes to these Hardy-Weinberg equations is identify the genotypes. Because usually you're either given the allele or genotype frequencies, at least one. And then you have to find the others as well as translate it into either an observed or expected genotype or allele frequency. So just to start, what are we given? We're told that 81 people have the condition. We're told their phenotype. What does that phenotype translate to?
It's little p, little p. Yeah, so. Oh, so it'd just be, it would be 81 out of divided by 300 to give us the genotype frequency.
So we have that many people with the recessive condition. What if it said 81 people have normal noses?
Then
Which genotype would that go to?
that'd go to the wait. If they had the other phenotype, it'd be a combination of the homozygous dominant and heterozygous
Exactly.
because they both exhibit the trait.
Yes, I should have waited to ask that question, but yes, you're perfectly right. So we have are given a genotype frequency. We're told the phenotype of a person or of people that only translates to one or is represented by one genotype. So that is the genotype frequency. 81 divided by 300 is divided by 3 to
3.27.
get 27 divided by 100. What'd you say?
Oh no, it's just I saw your math. 0.27.
Oh, no,
Sorry.
could you get rid of that?
Oh yeah, I can hide it.
Oh,
Uh,
I thought I waited that.
do you, I mean I can if you'd like.
Oh, no, that's fine. Actually, yes, please.
Okay.
Dang it, I'm
Do you want me
sorry.
to delete it or do you want me to just like?
Change it to white font.
Just can you the white?
Sorry, I didn't know it was up there. Okay, so how do we find the genotype?
Which one?
The ones that remain?
Oh, well, 81 divided by 300 is your, like you said, 0.27, so that's your recessive genotype. So then you can square root that and the answer gets you your recessive allele, and then you can subtract from one, get your
So what you're saying is we can't immediately find these genotype frequencies.
You can't find your, like your dominant or your heterozygous, but you can find your recessive. based
Yeah.
off.
From what we're given, we cannot find these genotypes. So just start with what you're given. It'll tell you something. And we can't make any connection to these just yet. Because you're right, these will be 119 of the 219 of the remaining individuals, but we can't distinguish between the two. So what we have to do is investigate the allele frequencies. So we start off with a single genotype frequency. It's very fortunate for us that we were given the recessive, because if we were given the genotype frequencies combined of, or the phenotypic frequency of people with normal noses, then we couldn't directly translate that to any
Okay.
single genotype.
Yeah, we'd have to assume and subtract. So if it said like 81 had regular noses, then we take 300 and minus that to get the homozygous recessive.
Exactly, yeah. We can only work with the homozygous recessive in this situation because it's the only one that correlates directly to a single allele. This one and this one doesn't. So we have to use this. Thankfully, we were given that. So with our Hardy-Weinberg equation, the way we get it, I can finally draw it, is if you have, for example, an allele P and an allele Q in an individual, similarly a P and a Q in their mate, you bring these two together, you have P times P, Q times Q, Q times Q, Q times P. What is P times P? What's 2 times 2?
It's 4.
Yeah, it's correct. You are adults. P times P is P squared. Q times Q is Q squared. And P times Q is PQ. This is also PQ. There are, so these are all the possible individuals, the only possible genotypes they can have, because we're only looking at 2 alleles in this population. So if we add them all together, so all of the individuals in this population, 100% of them, which we call 1 in statistics, is made up of the P squared individuals plus the PQ individuals plus the other PQ individuals, which combine to be 2PQ plus the Q squared. That's where the equation comes from. Only when you concern their gametes. So if you have, assuming that Q is recessive, P is dominant, we have a homozygous recessive individual. The homozygous recessive genotype comes from this association of gametes or alleles. Sorry. So this equals. That's right. This.
Okay.
So what is this? The single letter? It is the letter V.
I'm sorry,
What does it represent?
The dominant.
And what is this? What does it represent?
The genotype.
The genotype, exactly. And so when we see a genotype associated with these.
Okay.
So we have the observed genotype frequency, the one that we see in our population. And this is the ideal situation where all of the possible genotypes in the population are made up of this configuration of genotypes. And ideally, they'd exist in a certain frequency relative to each other. So, like Bernard said, we have our Q squared. Because they only have one type of allele, we can very easily translate it into the allele. That's a terrible way to say it. This genotype only occurs from having one kind of allele, so we can very simply reduce it. How do you reduce a square if you just want the Q?
I don't have the math readily available. Can I look at this? Oh, 0.52.
So we used our genotype frequency, the only one that we had available to make, to assume the allele frequency, or I guess find the allele frequency. Because these individuals, they only have this kind of allele. And so if, yeah. What do we do with this? So we couldn't do anything with this to translate it into that. We had to translate it into this. What can we do with this?
Subtract it from what? Why? Because that is one of the alleles, and there's two alleles in the population. So if you add the frequency of both alleles, it should equal what? Exactly.
So if we're only looking at two alleles, then it makes sense that 100% of the alleles that we're looking at are the first allele and the second allele. So if the second allele exists at a certain frequency, do some math. If one allele exists at a certain frequency, then the other one exists at the opposite frequency. So we have our one and we have our Q. That's how we get P. There was a much better way to say that. and I can't remember.
That makes sense to me.
We're only looking at two alleles. So when we find the frequency of one allele, and that is the frequency at which we find the allele in the population from what we know about the information we were given, the genotypes that we observed. So the genotypes that we observed give us insight into the frequency of one of the alleles that we're looking at, and we use that to find the frequency of the other allele. So now that we've found the frequency of the second of two alleles, we can now use this. Because it's true that 1 minus 0.27 or 1 minus this gives us this. So all of these together make 1. Now we have a way to distinguish between the two using the Hardy-Weinberg equation. So which one is this? What is this if we're looking under Punnett square? P squared.
Two PQ.
Why not just
There's two of them.
so these individuals are also having dominant at the first and recessive at the second could be these. Recessive at the first, dominant at the second could be these, but they both have the same genotype. That's why they're 2 PQ. So you have to consider these PQs and these PQs, and that's our 2 PQ. Okay, good. So we have that. Actually, no, let's not deal with that. So this is the ideal situation, but we don't know if we're in the ideal situation. Just a tip. Oh yeah, this was a much better way to approach the question. Find your allele frequencies. It's the allele frequencies, like that is the only accurate information that we can rely in these problems. Because there's like a circular pathway of trying to use this and then finding that everything works out perfectly. And it's not. But if you use these, this is just a derivative of the data we were given. So these are always going to be true, the allele frequencies. And then we need to use the allele frequencies to find these. So we can plug in P to P squared.
I had to. That one would be 0.231.
What is this?
That is the homozygous dominant frequency. genotypic frequency.
Now, why don't we use 2PQ? Actually, I think we can do it either way. Let's check that out. So how can you find 2PQ? How? What are those
Well,
ways?
one, you do the actual formula, you plug in your P, plug in your Q, multiply by two.
Or
Or
for 2PQ.
you can solve for Q2. So you have P.
Or you can just plug in.
You'll get
This one.
0.5 since you did 0.23.
Thank you. That makes sense. And what about this one? Actually, let's make sure.
It's a little off if we just stick to 2 decimal places, but if we go to a third, it'll be like off by 1,000.
These numbers will be different.
Just by like a
Like
hair.
instead of 0.5, it'll be 0.499. Since for P squared, instead of 0.231,
That's
stuff one. So
okay. Well, then it doesn't.
yeah, except for going because it's one.
Yeah. So
Zero, that's one. Zero.
Oh.
when we do the math, I think the key is to work with your allele frequencies.
Find the zero after the 5.
Fun.
2 6 big. There's a zero after the 5.
I don't believe you.
Zero is not significant. I put up a 5, so you only know one significant.
You need to.
Yeah, you need 2, so you need to add a 0.
This is
Your answer would be
chemistry's
wrong in the chem department.
perfect.
Just a
There's something I wanted to say. It's like kind of the key to. Does this make sense?
Yeah.
yeah.
Yes, I think I'm good. Now I'm good.
What's the process?
Well, you start off with your genotypic frequencies, which is what
Sorry, what if you're given an allele frequency?
That would even be less steps, wouldn't it be? You can just plug it in. You got it already.
Give me the tweet.
makes sense. That makes sense. I won't forget this on Friday. Other person? Oh, they're not done. She's gonna get them done tonight.
She said so?
That's what she told me.
Oh,
Can we work? I have this question. I don't know if you guys have this, but it's one that we did for exam 1, where we took the genotype of, we took 2 genotypes and they had children. And yeah, it was like, how many will have or what's the percentage of same genotype as the mother, father,
Yes.
phenotypically resembles.
Yes.
This, I have those up here. Just don't judge too much.
I have when I did that on the homework, and it's just so many fractions.
Yeah. Sometimes I overthink
It's just
things.
all fractions.
I spent like 2 hours after class with a group of 5 or 6 people. back
On
when
this
we were
question.
I was one of them.
Oh,
I remember that.
Like I get the, like I,
Maybe it wasn't 2 hours, but
It
it's
was
at
a
least
while.
an hour.
Yeah. It was like an hour 45.
So let's say you cross.
I have the question up if you like it.
Okay, so heterozygous there and there.
I wish I could zoom in. I'm so sorry. Freaking lockdown browser assumes that I can see. It's so small.
I'm very
Oh, I remember this question.
She asked about this one. Such color.
It's a big point question. So I feel like it's one of those questions that'll probably pop up. This is why I should use my,
Are you okay? I didn't hurt.
no, just made me sad.
When I see these questions, I just get overwhelmed by the amount of genes we have to look at.
Yeah.
I just look at it like allele by allele.
Yeah.
Perfect. Exactly. So
What are we trying to find specifically? Like the.
Oh.
first question is, what is the likelihood that any offspring will have the same genotype? Who's the first one?
Sorry, yeah, the first one's the mother, the second one's the father.
So likelihood of the same genotype.
That's pretty. You should probably like write it out. I see I might be the male.
Okay, yes.
Mom.
That's unfortunate that.
Yeah.
Yeah. It's like when I see pink and blue, I'm like, God, society.
Society.
Get with the times.
Okay, so the likelihood that an offspring, it's been a long time, sorry.
I know,
The-
sorry.
The likelihood that the offspring has any genotype. Okay, so the first thing that we have to consider is that these parents are going to cross. And when they cross, like they're going to be deciding through meiosis which of these gets passed on. Right? We just went through meiosis. And if we have for anaphase, then ooh, I actually have more colors. Oh, I feel like a fool. I had like, let's say that this is lowercase b and this is capital B. When we go through meiosis, we know that it's pretty much random whether or not, because these are eventually going to get to their own cells. And it's random whether or not this gamete or this gamete goes on to combine with another organism's gamete and then create the zygote that we will be investigating their gene. So what we can do, this is like very intimidating. Obviously, we can break it down. Or not obviously, but it's important to break it down. Because what Ma, in quotation marks, tells us that genes like aren't always connected together, that when they're in meiosis or metaphase, they can align at certain points.
Independent assortment.
The law of independent assortment makes it so that what happens at A, the chromosomes that contain A, or even the genes that contain A, aren't necessarily related to what happens at B. So the likelihood of any child getting this from their mother and this and this and this and this is just the likelihood of these.
Irrelevant.
Is these coming together to produce something similar? These coming together to produce something similar. So what we can do is just like break it down into a bunch of dihybrid crosses. So if you have, your famous AA times AA, what's the likelihood that a child has AA? Well, if we combine capital and capital, we get this. We combine capital to lowercase, we get this. And, you kind of just FOIL it out. You remember your elementary
FOIL.
math?
Foil.
Remember FOIL?
Never used foil. We didn't call it foil. We called it something else.
So if you have like 3 times 2.
We just said
plus
distributing.
Yeah.
7.
We never called it foil.
Okay, fair enough.
Yeah,
Um,
they
I don't
did
remember
that.
why
They did
we
that in
called it
calculus here, and like my second semester year. They're like, yeah, now we're going to foil, and I was like, what? And I just drew lines. I was like, oh, distributing. And it was like, what?
I didn't call it foil, I don't remember.
First, outer, inner, last.
Okay.
Yeah, yeah. My brain like houses things that are just unnecessary.
So from this simple dihybrid cross, these are the possible genotypes that we're looking for. And if we wanted this, the likelihood of getting it is 1 out of 4 because it's random whether or not this parent will give their capital A or their lowercase a. And same here, random whether or not it's capital or lowercase. So going to here, we have to think of what a parent can give. Firstly, what are we looking for? What we're looking for. We're looking for, see this? So it's
Yes.
So this
Sorry,
is
can
the
you
genotype. One second. So what is the likelihood that we find this? Why?
Because both of them have homozygous recessive, so regardless, they're always gonna give the little a allele.
Exactly. And that's the key to this question, thinking, what can these parents give? This mother only has a little a to give, so she will give it. The father only has a little a to give, so they will give it. 100%. Or
And
one. What about the next one?
1.
Right.
Same thing.
Same deal. They only have a capper. They only have a recessive to give, so they will give it. They only have a recessive to give, so they will give it. What about this one?
1 half.
Why?
Because when you do a cross, you have 2 big C, little c, 2 little c, little c.
The father only has a little c to give, so they will give it. The mother can give either a big C or a little c.
we need her to give a big C for it to be that genotype.
There are 2 possibilities. We need to have big C to get it, but it's random based on meiosis, which one they get. So it's just as likely that one is given as the other. So 0.1. What about the next one?
Same thing. 0.5. Same reasoning applies.
Which parent is the one that can give one thing or another?
The mother.
Exactly. What about E?
Same thing applies, except that instead of lowercase, we have uppercase.
Which one is the one that can give?
So in this case, it'd be the father.
The mother only has capital E's to give, so they will give it. The father has either, or they can give either dominant or recessive E, and it's random because of meiosis, which one they end up giving. What do we know about probability when we want? So in order to have this genotype, to have the condition at each of the loci. So if we know the probability of having the thing at each loci, how do we coalesce that into a single likelihood for that genotype?
We multiply.
Multiply with
Are the
Frequency.
individual.
yes, we multiply the individual frequencies together. And why do we do that? Why does that make sense?
Because it's the and. We need this and that to happen, so it's the probability of A and the probability of B multiplied together.
Exactly. And statements in probability indicate multiplication. And like a way to think about it, if you are unable to keep and or straight, is Bernard said it the other night that when you want and or when you have and statements, you want this and this and this and this, or else you're becoming more and more specific. And as you want or desire something more and more specific, it's less likely that it can just randomly be given to you. And so if something's less likely, the probability is minimized. And when we multiply fractions by each other, you minimize the value. So it has a 0.5 to 0.5, so it should be 0.125. Is that correct?
Yes. They died already. Didn't you get it like 2 weeks ago? He has more chance.
Oh, sorry.
Does that make sense?
Yes.
What's the next?
So the troubling one,
Yes,
it
let's
got
do the
me
trouble.
for that one specifically, but I'm glad we went through it because it's been a while. What did I just do with it? It was when it does not fit, when the kid does not phenotypically resemble either parents.
Phenotypically?
Yeah.
This is does not phenotypically
Either of the parents?
resemble either parent.
So what phenotype will this show?
The recessive.
Recessive,
What phenotype?
recessive,
That
dominant.
is-
Why?
Well,
Oh.
if we're thinking about it, all like normal.
Wouldn't it be zero?
Dominance,
It's
then
just straight
yes.
up impossible.
Not
That's what I
necessarily.
said.
This will show dominant. It's a heterozygote.
Yeah.
And we assume that capital is
Oh,
dominant.
wait,
over
never mind.
Oh,
I'm-
it's like a
I'm-
trick question, kind of. It's a trick question, isn't it? Kind of.
I'm not sure. I haven't done it.
Oh, the answer, sorry, I can tell you the answer. The answer is 50%.
So we can work our way there.
Okay.
This. These are going to be dominant. So what we want is just a dominant at the first, or recessive for the first two. And dominant for the last ones. So yeah, all we have to do is work our way through it. So to not phenotypically resemble either parent. So these are all going to have to be recessive. This is going to show as dominant. And what we can do is, so they will either resemble the parents or they won't. That makes sense. Right?
Right.
So if we want to find out how many of them don't resemble the parents, and find out how many of them do resemble the parents, subtract it from one. Because all children will either resemble or not resemble the parents. Hi, Safa, thanks for coming. Yeah. So if 100% of individuals either resemble or don't resemble their parents, and the percentage that don't resemble their parents is 1 minus those who do. So we can just find out the likelihood of the genotypes that resemble the parents and subtract it from them.
Okay.
So people who resemble their parents will have this little a, which we know is going to be 1. Little b, which we know is going to be 1. That helps us though. So if they do resemble their parents, they don't have their recessive phenotype, which only comes from a homozygous recessive genotype. And that's what we're looking for. So that's
Okay.
straightforward. Same with the B and T. C though. Who will resemble the parents?
All of them will.
Why?
Will resemble, because the mom is dominant and the father is recessive, so.
Exactly. The mother shows dominant, the father shows recessive. So it's not possible for them to not resemble the parents, because you have both conditions represented by both parents.
So I got 0. Like that's what I was understanding, was the answer was 0.
Yes, because we're looking for the ones that do resemble the parents first and then subtracting it from one.
So, so like it'll like differ. So like for example, like you see how the mom for C and D are both dominant, right? Like what if the son has dominant for C, but then gets recessive for D, right? So the mom has dominant for C and D, father has recessive for C and D. So if we get dominant C, but then a lowercase d, right, then it's different from both. So like they're
I understand that, but this is saying that the answer is 50%.
Mhm.
Will not resemble either parent.
I found them each like individually, like
So-
who would phenotypically resemble mom first, ignore dad, and then who would phenotypically resemble dad, ignore mom, and then added those together because it's an or situation,
Got it.
and
Okay.
then subtracted that from the one because it's the opposite.
Yeah, that's what. we're
Okay.
doing.
Got it.
So for the C,
I guess I.
for the C locus, the mother, let's once again just draw back, and if we can't find our way, we can use this guide. Only think, what can this person give? What can this person give? The mother can give either a capital C,
Oh yeah.
the mother can give either a capital C or a lowercase c. The father only has the lowercase c to give. So a child can either get this genotype, which resembles their mother, or they can get this genotype, which resembles their father. So it's 0 binary. So either way, no matter what they get, they will resemble one of their parents. And that means that C
Okay.
locus all of them resemble their parents, because that's what we're. All of them, because of what the parents can give, will resemble the parents at A. Same with B, and because of C, it's 50% likely that the mother gives the capital, 50% that they give the lowercase. And either way, they resemble one of the two parents, having homozygous recessive just like the father or heterozygous just like the mother. And in that case, 100% of them do resemble either parent. That's what the question's asking.
It is, but
So what I'm doing is finding the ones that do and subtracting.
so it seems like it's just the way the question was worded that it got me, because it's like it's in 4. Yes, it's 4. Do they not physically resemble mom or the dad? It's just weird.
Oh, okay.
Yeah, because the key that she has is 50%. So I guess if we take a step back, there's at the C locus, there's a 50% chance that it'll look like the dad, or 50% chance they look like the mom. I don't know. I think I'm gonna take a loss on that one.
For me, it was like, because it's, you're looking at like the specific combinations first. So it's the 50%. If you're just looking at the locus, there's 100% chance that they'll resemble either parent. But if you're looking at just like the total combination of genotypes, then you have to look at each parent specifically, and you can't look at either parent, if that makes sense. Like, so like with mom, it has to be, you have to be the recessive, you have to be the recessive phenotype, the dominant phenotype, what's labeled up there, but you can't, so I'm trying to think how So like you can't have the first two match mom and then the third match dad, if that makes sense. So you can't use that one when you're multiplying for who resembles mom, you have to use the 0.5.
I don't like this question. It's
So
okay.
rather than doing resemble either, you can break it down into resembling the mother and resembling the father. In that case, C is 0.5. D is also 0.5. Because the mother can get either capital to match this, which would resemble the mother, or they can get lowercase to match this, which would resemble the father, and therefore not resemble the mother. D is once again.
When how we get 0.5 from that is because E will be the same with the father in this case, because we're doing phenotypically, right? So the only real difference is the 0.5 between C and D. So for each parent, if you do that fraction, you will get a 0.25. And so then you would add up the like individual frequencies from each parent and you get 0.5, and then you subtract that from 1, and you get a 0.5 chance that it will not resemble either.
I get it. I just don't like it.
Yeah, I bet it would take like 30 minutes to do that question.
Yeah, I literally put 0 because I was like, that's not possible. Okay.
It's tricky, but if you break it down into the math, you do have to do some like math weaving. It's not nice and simple.
Yeah.
I like when the question will be
Yes.
built on each other, because then if you've already solved it, you can just add
Saves
those together,
time.
it saves time.
Accounts tomorrow, I have account. I gotta choose which ones to study for. Because I'm not studying for all 3 of them. And I want-
It's too much emotionally.
Yeah, I need to base off, tonight's gonna emotionally destroy me depending on what I get. I told Anita I got a 42,
What?
exam 4.
Exam for what?
Orgo?
You got yours
Well,
already?
no, I know I'm gonna get it. I know.
Oh,
I know I'm gonna get
I got a 42 on my exam for, or maybe it was like a 45 or something, but it was better than my
Orgo?
lowest grade, so at least my
Orgo.
lowest grade getting dropped.
wait, which class?
Orgo 1, so
With
different, but it's online asynchronous. I have never met the professor.
I don't know if that's like better or worse.
It's terrible. It's really bad.
You have to learn off the textbook and this too.
Oh, yeah, and he does not even match the content on the exams. So it's a fun little surprise what the final is gonna be like since it's not ACS.
That sounds off.
I will not be doing it again. So
You should watch the organic chemistry tutor or something like
Love
that.
him.
that's the only way, because like I've
I
gone
hate it,
through
but-
like the note slides and the textbooks for other exams, and we could take, like, other people have had the same issue. You can take like the exact source, like you can source the question back to the textbook and the notes, and it's wrong on the test. So it doesn't make sense.
Oh my gosh, I'm so sorry. That's so frustrating.
Worst case scenario, you have to retake. but you can take 0 or
Exactly.
you can-
I'm
you
gonna
can
be
do
taking
0
more.
or- Yeah.
That was her class got filled up. It's the only reason I didn't take it this semester.
I recommend Greg Petruncia.
Mhm.
He's my Orgo professor, and he's a really good professor.
Where does he teach at?
He's
Like, where is
online.
he? He's online?
He's in
Innovation.
Or is he in the? I'm so confused.
Where is it?
Apparently there's an Orgo
Innovation.
class in
Prince William
North.
County.
North, yeah.
Yeah, Prince William County.
Oh yeah, there's one on SciTech, and that's where Dr. Wood teaches, I think.
No, she teaches
Dr.
here.
Wood teaches here.
She might have started with it.
I've heard Eric wanted
Yeah,
to
SciTech Prince William.
take.
Yeah, there was one there. I was gonna do that, and then I failed Chem Lab 2, so then I couldn't take Orgo 1. So then I had to go over to or Gen Chem 2, which was fine because
I'm just hoping I don't have to retake or go lab 1 because I passed the lab.
Hey.
So that lab was fine.
That's
It was the
awesome.
lecture that's the problem. So
Dude, lab is rough.
yeah.
Lab is rough.
Chemistry
Lab's
lab's the
over.
worst.
Oh my
Lab's
god,
over.
I did
As a
better.
master's.
I did better in lab
Yeah,
than I did in lecture.
me too. Which isn't saying a lot, but it's saying something.
Exam.
I got a 67.
Yeah, that's a whole